Okay, let's get started. Welcome everyone. I'm Ganga Prasath. I'm a faculty in the Department of Applied Mechanics and Biomedical Engineering at IIT Madras. So this workshop, this online workshop, Ethology from Individual to Collective, is organized by Professor Danny Raj, who is in the audience, and myself. Before we get started, a couple of minutes about our department, and this won't take too long. So the Department of Applied Mechanics and Biomedical Engineering was founded in 1959. It's a seriously interdisciplinary department with faculties from 12 different backgrounds, 12 different departments essentially. And we are here, but doing interdisciplinary science, but broadly categorized in solid mechanics, fluid mechanics, and biomedical, primarily for legacy reasons. As you'll see, the kind of work that we do is pretty interdisciplinary. We are primarily graduate research department, and we have 230 odd PhD students and 120 odd masters and 80 MTech students. And this is the broad area of research traditionally that's done in the department, but with the kind of new faculty that we have, we are interested in cross disciplines that are not already present in this slide. And we encourage you guys to get in touch with us. We'll give you options by the end of this workshop by which you could get in touch with us. Just 30 seconds about us. So Professor Danny Raj, who is in the audience, is going to give the next lecture, is a faculty in our department. His broad area of interest is collective behavior, agent-based modeling, and equation discovery, which are some of the topics that he's interested in. He'll tell you more about it tomorrow. I'm Ganga Prasath. So my broad interest is in behavior quantification, robotics, bio-inspired robotics primarily, and decision-making processes. So you're welcome to go look at our website, and our lab is called Interface Lab, which is basically speaking to the interdisciplinary nature of our research work. Okay, so that's as much about the background of our department and about us and the kind of research that we do. More details, please visit the website. So the aim of the aims of the workshop are quite a few. One is to introduce you all. We have looked at your backgrounds in the sense through the form that we sent out. We see that you all have really diverse backgrounds. People coming from ecological sciences, physics, mechanical engineering, chemical engineering, electrical engineering, which is amazing because the field of behavioral science is really fascinating, and it is the breeding ground for people from different backgrounds to come and explore and exploit. And it is in many ways the right time, as we would like to highlight through the course of this workshop. There are a lot of interesting questions around us, which if you want to answer it seriously from a scientific viewpoint, you need multiple disciplines to come together. Okay, so the primary goal is for us to open doors to this fascinating world. The second, like I mentioned, is to introduce or highlight the interdisciplinary nature of problems. And through the workshop, also introduce some of the modern techniques that are becoming staples in this field, and maybe a few applications, either in robotics or in network design or algorithms. If you're interested in that kind of thing, we'll highlight those through the course of this workshop. And lastly, one of the things that we have realized, as graduate students, is that workshops tend to be much more effective when you put in the time after the lecture itself. So that's why we have a couple of mini projects, which we find exciting, and we hope you do as well. So you'll have to spend a few hours after the workshop, and there is a small reward at the end of it. One which you already know, another which hopefully you'll know by the end of this lecture. Okay, so those are the broad aims, and this first lecture is going to be on Introduction to Ethology. So we'll get there. Hopefully, by the end of this lecture, you'll have some idea of ethology. So the primary resource that I think people in the field all respect, is this book by Alcock and Rubinstein. It's called Animal Behavior. It's I think it's like 1,000-page book, which is like the reference book for a lot of things that we'll be talking about, and we'll literally be just scratching the surface of this book. And another really nice book which gives you more quantitative picture is this book by a set of scientists who are all pioneers in the field. It's called Self-Organization in Biological Systems. Both of them are serious books, but very readable, approachable. Those are the science books. There is also good pop science, which I do encourage you to go and look at. There's a very fascinating book that came out a couple of years back called An Immense World. It tells you it talks about how the animals perceive the world, similarly animal-wise, and then the mind of the bee. These are just a few examples. I think there are a lot of books out there, but if you're uninitiated, I think these are really good books to get into it. Okay, so one on serious science and other on stuff to keep your brain picked. So this talk is partitioned into two parts. So there's the first act where you'll get introduced to history of ethology, what is behavior, the underlying mechanisms, and a classical paradigm called the Tinberg and State triad. The second half, which is going to be on the techniques that are used today, actually, literally, it's becoming the bread and butter of behavioral science, which is using machine learning based techniques for tracking and behavioral quantification. We'll get to that. So we'll get started with Act 1. So what I'm going to do is show you a series of videos. I'm going to describe what's happening. It's a puzzle, and I want to ask you guys by the end of this series of videos what you think are the takeaways or the common things that you find from these videos. Okay, so that's how we're going to go. Okay, let's start with this video. So this is a salamander. The salamander is this is in a lab, of course. So it's walking from let's say land or solid substrate to a fluid medium. Let me play that again. And you see, initially it's walking, and its gait naturally transitions from walking to swimming. And then once it finds land, it again starts walking. Okay. At least I was very surprised when I first heard that brain was not at all involved in this gait transition. This entire mechanism by which it underwent this gait transition was purely a reflex-based transition. As in, none of the brain cells were involved in walking or and then undergoing transition to swimming. Okay. So what was found later on was that there are these set of neurons called the central pattern generator, which drive this transition. So I'm not going to give you too much details, but the point of this is for you to be picked and then go and read about this, or we can have a discussion by the end of this talk. Okay. So the first point is this gait transitions have nothing to do with brain. Okay. This was this paper which was published in Science in 2007 was by a robotics group that replicated this behavior. So you can see here they built this robot that has no brain, and just out of reflexes, which is to say just local sensing and response, it's able to replicate this gait transition. Right? So this is a robotic implementation of this gait transition without a brain. Okay, so that's the first video that, or rather first two videos that I want to show you. I'm going to just continue along so you can see that they've replicated this. And the second is this beautiful butterfly, which is very famous. It's called the monarch butterfly. So the monarch butterfly attained its status of fame because it undergoes it goes on the serious migration from Mexico to the United States and back to Mexico. But the interesting bit is that it's not the same butterfly that completes this migration, this migratory trajectory. So what happens is that the monarch butterfly, by the time it comes back, it's essentially the great grandchildren of the first butterfly that left Mexico that comes back. So which is to say that the monarch butterfly that is starting out from Mexico, getting into the US, is basically its offspring is migrating through US, and its offspring is experiencing different seasons, and its offspring is getting back into Mexico. So this entire trajectory people found was very, very reproducible, in the sense it had a prototypical trajectory it takes along this migratory path. So this must really blow our mind, because how is it that the great-grandchildren of these butterflies are able to remember this path if nobody is actually telling them how to go about it, right? So there are several hypotheses. One is that it might be encoded in the genes. So that's the second bit of a story that I want to share with you. So I'm going to go to the next one. Okay, so the next is again a lab experiment. So this is basically a collection of our favorite housefly. So we all have enough of them in India because of tropical climate. So here, the dots that you see are a collection of housefly, which is confined between two plates. And you have white background, which is just light. And what you're going to see is a smoke that's going to come from left to right. Okay, so let me play the video. So these guys are put to sleep, they wake up, and then you switch on the smoke, and then this is what you see. One important thing is that this is slowed down initially, and it's going to speed up soon. Okay. So what you see here is that these housefly use some interesting strategy to go from wherever they are to the source of the smoke. Right? The smoke is nothing but apple cider mixed with some color, and then you just push this apple cider out of a bottle through this experimental setup, which is between two flat plates. Right. So this is the third bit of video that I want to show you. So it's seemingly simple, but fun experiment, I would say. Okay. So next is a super interesting insect, which is a desert ant. So desert ant, again, is one of those ants in the Hall of Fame. It's called Cataglyphis. So here is a trajectory that a desert ant takes from its nest to its food source and then back. So I'm going to tell you. So desert ants, as the name suggests, they live in deserts where the temperature can go from, in the early mornings, maybe from 0 degrees to as much as 70 degrees, right? And as you might know, ants, from the video I had shared during the Google form, ants follow pheromones. That's one of the primary ways by which they track food, right? Whereas in desert, there's no way by which pheromones could survive, because if the sun is heating the sand to 70 degrees, these pheromones are going to just vaporize, right? So there's no way of encoding pheromone information in the sand. That's one. And the second thing, as you might know, the sand itself is highly dynamic. Over a matter of a few hours, the topography can change really rapidly. I was really blown away when I first heard about it. This is the nest of this desert ant. It leaves its nest. This is the outbound trajectory. It's going out, exploring, looking for food, right? It's doing something like this. And this is 10 meters. So by the time it has reached here, it has gone off on a length of 1,500 meters, 1.5 kilometers it has traveled from the nest, right? This is its home, this is the nest, and it has gone somewhere and found the food, which is path length 1.5 kilometers from its home. You know what it does after finding the food? It just walks straight back home. Right? This small insect, which is less than a centimeter long, is able to traverse 1.5 kilometers and just walk back home. Right? So this must be, I mean, really superhuman, right? To be able to do this. So this led to a series of very fascinating experiments, primarily led by Wiener, who showed that ants have, I'm not going to tell you the full story, as with other stories, I encourage you to go and dig up deep. One of the things that they found was ants have two kinds of sensors, if you want. One is a odometer, which is a sensor, let's say, that counts the number of steps. And the second is a sensor that can measure angular velocity or angle. So one is something that measures this step. The other is something that measures angle or the orientation. Okay, so I'm not going to tell you the full story, but this experiment actually led to people looking for a mechanism by which ant might be doing this. And there is one more very, very important information, which is that ants' eyes are sensitive to the polarization of sun. Okay, so that's another very important information. Okay, anyway, so those are the details, but the experiment is what I want to highlight. Ants have the capacity to walk really far and then get back. Okay, there's something super cool that came out just last week in this very, very good journal called PNAS, where they showed that ants can solve what is known as a piano movers problem. Okay, so these are the collective of ants. This is an ant collective that's trying to transport this T-shaped object from the left chamber onto the right. Okay, so they somehow trick the ant to do this task. Okay, you see that they're trying to find a solution. And I'm going to tell you something that these experimentalists did right after this video ends. That's it. These guys have actually solved the ant movers problem. This paper, you know what they did? What they did was they put 11 humans. They made sure they can't talk to each other either by sign language or via they can't speak, basically. And they showed that ants actually are more efficient at solving this task than humans. Okay, so the role of language seems to be really, really important. Okay, so that's another story. The next story that's coming up is that of bees. So here you have essentially a queen bee. A beehive has a queen bee. And the queen bee, so depending on the species, there is hierarchy within the beehive. So the queen bee generates what is known as a queen retinue pheromone that attracts worker bees. So this queen bee is inside this small capsule, and then it generates this pheromone which attracts a few of the workers. And these workers generate what is known as Nasonov pheromone that is then fanned. These guys are not flying actually, they're using their wing to flap and then transport this pheromone. So this pheromone is a recruitment pheromone, which is to say it will attract other workers to wherever they are. Right? So let me just repeat. So there's a queen bee which generates this queen retinue pheromone, and then that is sensed by a few worker bees. Those guys further generate Nasonov pheromone, and then it's like a cascade, and they're able to recruit other workers, maybe to help the queen bee get out of the capsule, let's say. Okay. The next story is again a super, super fascinating story. So this is a story from the 1940s, late 1940s. by this psychologist called Tallman. So this is the experiment that he conducted. So what he did was he took a rat, and he built a maze. So he let the rat explore the maze for, let's say, a few hours. I think he did it for like 11 hours. I don't quote me on that, but yeah, he let it explore the maze for a bit, and then he placed food in this food box. Okay, so he left the rat here, and the goal of the rat is to find the food, right? You starve the rat, and you have let the rat explore this maze. Now you let it find its food. So what he saw was, it took a particular path to the food source. Then what he did was he took the same rat, put it at a different location. What it did was it did not do any exploration. It just walked right to the food source. Okay, so let me repeat. So there was an exploratory phase where the rat got familiarized with the environment, and there was a trial phase where he tried to leave the rat at different locations within the maze, and what he found was the rat went to the food source without any exploration. Okay, so this led to a paradigm of learning, which is associative learning versus map-based learning. Associative learning, if the rat had used associative learning, it would have tried to use the same strategy it did in the first time, right? After it had explored, let's say you leave this rat, it goes via some trajectory, and then it goes to the food. If it is associative learning, if you leave it at a different location, it's going to actually try the same strategy. Because if the rat doesn't have an idea of this entire maze or a map of this entire maze, it might just use the same strategy. But what he found was, during the exploratory phase, what he proposed rather was that the rat is forming a map of the maze, and because it has formed this map, it knew precisely where the food was. Okay, so in fact, there are a series of experiments that people have found, people have done, and people have found. Actually, one of them led to a Nobel Prize in 2015. Was that if you want to form a map, you need to have what are known as place cells, which give you a sense of place. And if you want to place yourself, you need an underlying coordinate system, which is what are the grid cells. So there is an underlying neural mechanism that helps you perform map-based navigation. Okay, so that's the other story I want to show you. A couple of really small, quick stories, and then I'm going to ask you guys a question. I'm going to ask you guys an answer to the question I started out with. So this is again a very famous experiment, which I think went viral in the social media. So this is a chimpanzee which can remember the sequence of numbers that are flashed. So let me tell you what the experiment is. So initially, this chimpanzee was trained to remember the scene that is flashed, and then the numbers that are flashed, and then it has to sequentially click those numbers. So let's say 1, 2, 3, 4, 5 is flashed. It had to click 1, 2, 3, 4, 5 in the increasing order, in the ascending order, based on the location that it was flashed in. So you'll understand what I mean. So this guy it was flashed and then it was hidden behind that white square. It was flashed literally for 50 milliseconds or something of that sort. So this chimpanzee becomes super famous after this paper from 1985. The chimpanzee, it's called AI, and humans failed miserably in this task. And chimpanzees, so AI was successful 90% of the time. Okay, so this led to a lot of questions in neuroscience. I'm not going to be spending time on that. one of the things that is hypothesized is that we had to trade off with the short-term memory which is required to perform such a task because we had to use that part of our brain, which is the prefrontal cortex, for language and decision making, both the hippocampus and prefrontal cortex. Okay, so the last story is about and I think a behavior workshop can't go without the audio of curious, which I believe you must be. Please go and continue watching this fascinating short film or series called The Incredible Life of Ants. Okay, so that's the last video. So let me just ask you guys the question again. So what you think was common that helps you essentially define what behavior is? That's the question that I'm getting at. So these are all examples of an organism performing a particular behavior. So what are the common things that you think are required, that are performed by these animals that are required to define behavior itself? Let me go to the answer. So there was motion in all of the videos, right? So movement somehow seems to be extremely fundamental to characterize. So these are all the ways by which we can characterize behavior. And as Akriti pointed out, there is this notion of a task, right? So that the organism is trying to perform. And a crucial part of this is this process of decision making. Right? This is probably the most important aspect of the behavior itself. And there is something like memory is useful in representing the task. So there is an aspect of representation. Associated with that representation is this notion of complexity. How complex is the task? I mean, it's still very vague, but there is this notion of representation, and associated with that representation is this notion of complexity. And of course, the examples that we saw had both individuals and then the collectives. Some of the tasks that the individuals performed maybe were difficult for collectives. For example, in the ant case, some of the tasks that the ant collective performs, clearly an individual cannot perform, right? So these are all aspects that define behavior itself. We can maybe at least come up with a definition, which is that behavior is the science of decision making, and that most often results in motor actions. Okay, so this motor action is driven by this fundamental loop, which is sensation, perception, and action, right? You sense the environment, you process that information that you sense, and then make a decision, and then that results in action that eventually transcends into motor action or movement, right? So this is a working definition of behavior, and there is an important definition, distinction between human behavior and animal behavior. There is a very, very important distinction, and that has to do with language. So that language is very fundamental because you can't ask an insect how they arrived at a decision, whereas you can ask humans how they arrived at a decision. So that is one of the classical distinction between animal behavior and human behavior. And that for that particular reason led to psychology for the longest dealing with behavior, going back to, you guys know who this person is? You can raise your hand. I know you guys would know this, and I know you guys would know this. Who are these two gentlemen? Okay, so this is Pavlov from the Pavlovian classical conditioning. This is Skinner. Okay, so this is Pavlov, Freud, and Skinner. So they are some of the most famous psychologists from the classical era. And then there is Chomsky who's a linguist, but who has also extrapolated some of his findings to behavioral science. So the history of behavioral science was initially led by psychologists and cognitive scientists. And Skinner actually was one of the most important people to talk about what is known as operant conditioning, which is to say certain behaviors which lead you to positive rewards are the ones that are reinforced. And this inspired what we today know as reinforcement learning. Okay, so the algorithm reinforcement learning goes back to Skinner. And of course, Pavlov came up with this idea of classical conditioning, and Freud came up with archetypes on how people make their decisions. Okay, so that's the background history and the distinction between human behavior and animal behavior. But how is this behavior encoded? Right, I think one of you alluded to it being physiological. But this question itself is a mechanism question. So if you want to ask where and how is behavior encoded, so that if you want to answer, you want to understand the mechanism by which behavior is encoded. And there are actually three ways that are possible. One is the genetic basis, which we saw is one of the hypotheses for the monarch butterflies. Second is the neural basis, which we saw in a variety of examples, starting with salamanders to ants to rats. The third is a physiological basis. Physiological just has to do with stimulus response type behavior, where a particular organ performs that, for example, in the National gland. So it's a physiological basis. Something that's hidden in the background and plays an overarching role is actually the environment. So if you want to explain a particular behavior, so the mechanism by which it works can fall under any of these three categories. Okay, and there's a very, very important notion given our understanding of evolution. There's this important notion of hierarchy and subsumption. Okay, so I'm going to explain this with a particular example, which is again super fascinating, and it has applications, in robotics and whatnot. So this particular example that I'm showing here is for the task of navigation. So this is again one of the classical problems in both animal behavior and in robotics. I don't know if you guys have heard of this problem called SLAM, spatial localization and mapping. Anyway, that's a classical problem in robotics, which can be directly mapped to this particular aspect of navigation in animals. Okay, so insects broadly, I heard people are interested in bacteria. So you can call primitive organisms perform navigation through very simple mechanisms. The simplest mechanism that we know is called locomotor guidance. This is either climbing up gradients. Let's say you have pheromone trail, you have chemotaxis, which is done by bacteria to climb up gradients in concentration of chemical concentrations. And these are all egocentric. Egocentric, which means that it's all from first person's viewpoint. So when I just pursue my neighborhood, my perception capabilities are limited, and I just perform this simple climbing up the gradient or climbing down the gradient, right? So obviously, you need a physiological basis to perform this task, or rather perform this motor action. But it's an extremely simple strategy by which you can navigate the world, which is what is done by bacteria, also done by other primitive organisms. Maybe I actually do. Yeah, it's a lot of the processes within our human body. The cells also use these simple strategies. The next level, or in terms of complexity, is what is known as path integration. So path integration is the strategy used by desert ants. It can remember its home by performing what is known as path integrations, where it's keeping track of its home. Wherever it is, it has what is known as a pointing vector or homing vector, which points towards the home. So you need a slightly more complex neural capacity to perform this path integration, this mode of navigation called path integration. And one more level of complexity is what is known as landmark-based navigation. Landmark-based navigation is simple. We use it all the time. Oh, I reach this street, I take left. That's a landmark. So, oh, I go near this tree, go towards my right. Right? So that's a simple landmark-based navigation. Again, that's one level more complex. And you can see we are building up to this whole idea of map-based navigation. Map-based navigation or route-based navigation is the most complex form of navigation that exists, where we are able to form a map of our environment in our brain. When you're able to do this, you have gone from this egocentric view to a geocentric or an allocentric view, where you now have a clear notion of the environment and you can place yourself in that map. Right, so that's the different layers of complexities, and this is hierarchical in some sense because the circuitry that's required to perform this task is more or less conserved, which is to say an ant can perform, or let's say a rat can perform both pheromone-based navigation and also it can do path integration and it can do landmark-based navigation, right? So there is this hierarchy of strategies, and if you can do a complex strategy, you have subsumed all other lower forms of strategies, right? So that's this notion of subsumption, where complex strategies subsume all lower strategies. Okay, so in fact, this map-based navigation was hypothesized in humans, and it was found just last decade. Right, we do have a specific set of neurons to perform map-based, route-based navigation. So there is a clear underlying neural basis for this strategy. Okay. This is just to explain that there is this notion of hierarchy in when it comes to behavior that is backed by one of these genetic basis, neural basis, or physiological basis. Okay. So the last theme before again we open for questions is this classical paradigm of Tinbergen and tetrad. So we have seen several examples, if not several, a few examples, and then we have seen how to characterize behaviors. Now, if you want to ask a question in behavior, are there general class of questions that you could ask? So this was what Tinbergen came up with. So Tinbergen is one of the founding fathers of this whole field called ethology. So Tinbergen is also a Nobel laureate. You should go and read about his work. So Tinbergen and Carl von Frisch, Nicola Tinbergen and Carl von Frisch shared the Nobel Prize. So Frisch has done a lot of work on bees. He is the one to figure out this bee survival dance, the mechanism by which it's performed, and things of that sort. So Tinbergen, in his classic paper on aims and methods in ethology, he talks about 4 ways by which you could ask questions in ethology or in behavioral science. One is a question about function. Why is a behavior performed, or why does an organism perform a particular behavior? How does that provide better fitness or better efficacy in terms of being able to perform a task? So that's the function question. The second question is causation, or the mechanism question. What causes a particular behavior to be executed? What is the underlying stimuli response, and what is the physiological basis for this? The third is an evolutionary question. How has this behavior come up over evolutionary time scale, which has to do with adapting to local environment and things of that sort. The last question you could ask is a developmental question. How does that behavior change over the lifetime of a particular organism? So these are all in fact, you can ask any question in behavior, and it's nice that you will most likely be able to box that question in one of these 4 themes. So Tinbergen was, obviously a serious thinker, and he came up with this tetrad, which is a very sort of rubric to think about behavioral questions. Okay, so that's where I'm going to end the theory part of this, and we want to move to Act 2. So this is the end of Act 1. I'm going to again open up for questions. Okay, so good. So we're doing good on time. So I'm going to go to the second act, which is the technique part. So we have seen broadly the kind of things that Let's say, a lot of stuff that I've cited is quite contemporary. What we'll see in the second half is even more recent. So quantifying behavior has made this whole behavioral science a serious endeavor. So, initially it was psychologists, they were debating on flimsy grounds, I would say, because it was all based on qualitative assumptions and premises. And now it's becoming hardcore science because we are making it more and more quantitative and predictable and reproducible. So in order to do that, there are a few techniques that have come up, and as with machine learning, everything today, machine learning has an important role to play in this. Okay, so if you want to if you want to quantify behavior, you have to first identify what are known as motives or syllables. Some people call it behavioral syllables or motives. So this is a simple example where we saw that this housefly, is trying to reach this source of smoke, source of apple cider smoke. So if you want to quantify, you can define what are known as motives. So in this case, this motive could be encountering smoke, just sitting, or walking. It can't fly. So you can say these are the three motives, and you want to detect them, and then see from this motives if we can say something about how it's making these decisions. So that's the ultimate goal, right? We want to find out how this insect is making its decision, how it's arriving at a particular decision. So in order to do that, of course, this is a purely sort of anthropomorphic or a human way of looking at it, because the motives are defined by you. So you are defining the motives. So you have to define the motives, you have to identify the motives in the videos, and then classify them. So, and then of course you can look at the data that you have from the videos, and then see if you can find how the agent might be, or the insect might be able to make its decision. Right? So in this particular example, this is from as you can see, from a paper, very recent paper. This is the statistics that they have got. So here they've done, I don't know how many experiments, but this is the trajectory of all the insects, 400 odd insects, and this is the encounter that they have seen with the smoke, the insects, the housefly with the smoke. You can plot its orientation. If you track the insect, you can find the orientation and see how this orientation is associated with the speed, its motion, and then so this, I forgot. So this is the signal. I'm sorry, I'm blanking out. So you can basically quantify its orientation, its speed, and I'll figure out what this signal is. Might be in the next lecture, I'll tell you guys. Okay, so this is one way by which you can quantify the behavior of this insect. And then, of course, you have to look at this data and find how it's making its decision. And one of the findings that they did is like a stimulus response behavior based on the orientation of the smoke and how it encounters it. This guy is performing this action. A more general way of quantifying behavior is using this method called the ethogram. This technique called the ethogram. So ethogram is, again, you have to define a motive for the organism, and then as a function of time, you find how this motive is undergoing transition. So on the x-axis, this is an ethogram. This is an example of an ethogram. On the x-axis, you have time or frame number from the video. On the y-axis, you have the behavioral motive. So here, the researchers have defined 4 motives: scratching behavior, digging behavior, body grooming, face grooming, and background, which is actually they're not able to detect. So that's the background. Right? So ethogram is a much more general way of expanding from this encounter versus time to, let's say, time versus a particular motive. So here is a video example where they have, this is basically the same example, but in video form. So this is one way by which you can quantify behavior, of course, you have to look at the data to find out how the organism is executing this particular task. But this is one of the fundamental ways by which you can characterize behavior. So earlier, the classical way of using ethogram was, the researcher used to sit in the wild with a notebook, note down the time, and then find out what the organism was doing. Literally, there are so many examples of this where people sit and just note down what the organism is doing. So now, with computer vision, and I'm going to come to that in the next few slides, we can do much better. Okay, why is this useful? You might ask, what am I going to learn by just looking at this motor action or this motive and the transition between different motives? So this is what is possible. So you have this observation, which is a recorded video of the organism performing a particular task. You can perform what is known as pose estimation. Pose estimation is nothing but you determine the pose or the shape of different parts of the body as a function of time. So pose is nothing but, what pose am I holding? So my finger is open, my fingers are open, I'm keeping them closed. So open is a pose which can be characterized by the position, or rather the coordinates of my bones and the orientation of my bones, right? If I give you the collective information of all skeletal structure, I can characterize open hand versus closed hand. So pose is nothing but the three-dimensional data associated with a particular part of your body. So that's the pose. So from the observation, this important task of pose estimation needs to be performed. If you assume a model for this pose, then you can also infer the kinematics. Kinematics has to do with forces and moments that are applied by the muscles to hold a particular pose, or let's say undergo transition from one pose to another. Is that clear? So this is where the physics comes into play. So the pose estimation is a vision problem. You have the video, you estimate the three-dimensional pose, and from there you can determine the torques and the forces that are required to apply to hold a particular pose, or let's say in a dynamical case, move from one pose to another, which is a kinematics problem, or strictly speaking, it's an inverse kinematics problem, which is something that roboticists are very familiar with. And from that, along with neural information, let's say you have ways to probe the neural circuitry, then you have everything that you require, right? So you know which part of the brain is involved in holding a particular pose, what is the role of that neural firing in order to actuate a particular neuron, a particular muscle, and how is that affecting the three-dimensional pose is all that you need to characterize a behavior. So there is what is known as a descending path, and then there is what is known as an ascending path. So the ascending path has to do with sensory information. So that is missing in this case, but this is where behavioral science is at. So a lot of people are now trying to understand how to bring in this sensory information into this loop. So this is the descending path where the brain fires, and there's a descending set of pathways that translate to motor action. Okay. So this is the state of the art by which you can quantify behavior, especially the neural way of looking at things. Okay. As you can see, if you want to quantify behavior, it's a highly interdisciplinary problem. You need expertise in computer vision. You need to develop novel numerical methods in order to quantify these or to solve these inverse kinematics problems. You need expertise in biomechanics, because you see that muscles and the neural circuitry are involved. And of course, it's a problem in neuroscience if you want to probe the fundamental neurons that are involved in this particular behavior. Okay, so but I'm going to talk primarily about the role the computer vision and maybe a little bit about numerical methods, how that has actually changed this entire landscape. So if you look at this, if you can perform, forget about the neurons, if you can perform this particular part, observation, pose estimation, and solving an inverse kinematics problem, you know a lot about the behavior itself, right? So that's one way by which you can quantify. Of course, this is much more, it's going to give you much more deeper insights, but of course the experimental barrier is very high if you want to probe the neurons. So a lot of it changed when machine learning came to the fore. This was in 2017, essentially between 2012 to 2020. This whole Google came up with this package called the DPose kit. So this is an example where they're able to track the three-dimensional pose from 2D videos of this entire collective. So this led to a almost a revolution in behavioral science, because people are now able to get the pose data and then also get the inverse kinematics. You can do the inverse kinematics and get kinematics data, information about forces and torques. Okay, so these are slides from Alex Mathis. I'm going to talk about a package that he and his colleague developed. It's called DeepLabCut. That's where we are heading towards. So deep learning, the advantage of deep learning when it comes to pose estimation is that the amount of data that you require is extremely less compared to traditional techniques. Let me explain. So what you do in traditional deep learning is you train a neural network for a particular task with some hand-marked data, and then you supply it with, let's say, similar looking data. It's able to perform this task that you've trained on. So it's as simple as that, right? It's a function approximator that performs a particular task that you've trained on. So you, in the case of pose estimation, you give an image, push it through the neural network, and you get pose data, right? So the advantage is that with very little amount of training data, you can actually get the pose information. This is because of what is known as transfer learning. So transfer learning, the idea of transfer learning is that you train a neural network for a particular task, and use it for a different task by training the same neural network that's trained on a different task, but with this new task, right? So in transfer learning, you take a network that's trained on a different task, and you ask it to learn a new task, which is not radically different from the initial task that it was trained on. So as you guys know, the problems in images, people have just trained enough number of networks. This is an example, ResNet 50, which was trained to detect cats and dogs and all kinds of animals. So what Alex Mathis and his colleague did was to take this network and train it to estimate pose, which is not far from what other people have done at Google and other places. So, yeah. I think I should show this slide first. Okay, so what they did was, so this is this package that they developed called DeepLabCut, where you have, let's say, a video, you create a skeleton of the part that you want to track. So in this case, this is an example of a rat which is holding a lever and it's trying to move the lever in order to perform a particular task. So that's what the researchers wanted to track. So what you do initially is to mark or label the points that you want to track in this image, build a skeleton out of the marked points, train the neural network, which is a ResNet 50, which is trained on a different task, and then you train it to give the pose information, right? So you give this video, you give the output as this skeleton, right? That's how you do training of a neural network. And once you train on this with very few number of images, in this particular case, it was of the order of 100. Just train 100 images, and this is actually the output you get. This came out in 2018, and this already has, I think, like 2,000 citations or something of that sort, because this paper was really a revolution in this field. You can see it translates so well, right? You just trained a couple of hundred images, which takes you, maybe half an hour. And then that's it. You're able to do experiments where you're able to estimate pose For videos, this is an example problem. There's a code that's available online. I think this video has like 5,000 images or something. So, and it translates well for different experiments. The nice thing about CNNs, convolutional neural networks, is that it's scale invariant, it's translation invariant. You have the same rat sitting at different location with different hand size and different number of digits. It's able to do all of that. Okay, so that's what DeepLabCut did, and DeepLabCut was not the only thing that came to the fore, in the last few years. So there are other packages developed by other groups. Here's an example. So this was developed at Harvard and EPFL. This is developed by a group in Princeton, Sleep. This is another package primarily meant for tracking collectives by a group in Konstanz. But I would say the most widely adopted software is DeepLabCut, and it has several plugins now. People have built several plugins for this DeepLabCut. Okay, so this is the pipeline. I think, as you guys have already seen, so you have to perform training. You have to label the model. You have to train the model, and then you can use it to evaluate to get the post data. So people have tried to use this paradigm to also generate the ethogram. So ethogram, as we have seen, involves a couple more steps. One is defining the motive. The second is identifying, which you can do with neural network. The second task is classification. So again, you guys might know classification is a task that machine learning aces. So people have integrated both of them, and they've come up with a deep ethogram. This is a paper from 2021. I'm not going to show stuff from this, but if you're interested, you're welcome to go and look at this. But this was all possible primarily because of this entire pipeline that people have built in Vision. So computer vision community has built a lot of tool sets that people in the behavior community are utilizing to understand different aspects of behavior. And as I said, there are several tools that are built on top of DeepLabCut that, for example, you can solve inverse kinematics with DeepLabCut data. There's a plugin for that. Okay, and as I showed, there are ethogram plugins. Okay, so that's the place where we're going to shift to demo. Before we shift to demo, I want to just take a brief pause and ask you guys if you have any questions. Okay, so DeepLabCut is a very, very well documented software. That's one of the reasons why it has had such wide adoption. So the installation procedure is very, very simple. If you know Python, which you ask me, you must, given the world that we live in. So if you know Python, the installation itself doesn't require you to know Python. So there's a GUI. I'll just walk you through the GUI. So the way you do installation is to install Anaconda, and then inside Anaconda, they have given you a YAML file. There's an environment, all the environment variables required to use DeepLabCut are in this file. Once you download this, you can just use Conda environment create. YAML, it will install a lot of packages that are required, and you're ready to go. So once it's really a matter of a few minutes after you install Anaconda. Okay. So as you will see soon, there is a mini project related to DeepLabCut. So unfortunately, if you need a certificate, or in fact, I would say if you want to, have a wholesome experience of the workshop, I would, or both, I can speak on behalf of Professor Danny as well, that you please take the assignments or the mini projects seriously. I'm sure these are fun projects. You'll enjoy doing it. You can share it on Instagram or wherever you are. Okay, so please install the software. And once you're done installing, you can just run this command, python-m DeepLabCut. Okay. So if you run that, it's going to open up this window. By the way, so there's a question of how much time it will require for the project. It max 3 hours, not more than that. Okay. So there's whatever I'm going to show today is also provided in a nice tutorial by the DeepLabCut community itself. I'll share the link. But nevertheless, I'll quickly walk over this so that you also see it in action. Okay. So this is the interface that will open up. You can create a new project. So before I even do this, so the point of this is to perform post-estimation, right? I have a video and I want to perform post-estimation. So the example video that I'm going to use is one of the classic examples that's used by the DeepLabCut community. It's this mice moving along a track. It's supposed to track the trail, but I think this, yeah, you can see. Okay, so this is the video, and I want to track the body pose of this mice. Okay, so it's quite a long video. You'll see it performs ridiculously well with, I trained this in the morning with, I'm not joking, with 16 images. And I'll show the result that it gave me with just 16 images. Okay, so that's the point. That's our goal. So we want to track the mice on this trail. Okay. So after installing DeepLabCut, I start and then I give a name to the project. Let's just call it test or something like that, session, and give it my name. So here you see there is an option to do multi-animal tracking. And I'm not going to show this, but you can use this. So I'm going to browse the folder and pull up this place where this video is. So I think that feature is not there. So this is the video that I just showed. So I'm going to just check that and click create. Okay, new project created. So it's a very minimal interface. Actually, the interface also is built on Python, so it's not super fancy. So once you do that, you see there is this Python session that's created, and it has created a set of folders. So the DLC model, DLC is DeepLabCut. DLC model is where the trained neural network is available. So the label data, which we will see how to label, is available in this. And the training data set, the data that's used for training is available. This is the place where the video is kept. Actually, as of now, you can see it has just kept the shortcut. There's an option to just copy the video also. I forgot to check that box. Okay, once you've created this, the important file is this file called the config.yaml. So this config has details about the location of the videos. If you want a particular video to be cropped, if you want a particular region of the video to be used, you can also specify that. If you can define the body parts that you're tracking, name of the body parts. You can also define the skeleton. Okay. So once you have markers defined, you can construct the skeleton by just giving 2 nodes. Right. This is all 2D. You can also do 3D data. I'm not going to talk about it. You can check out their YouTube channel. So in 2D, if you define 2 points, you can connect by a straight line. And if you give a sequential segments, you can construct a skeleton. Right? So that is also possible. So I'm going to give you the most minimal version of it. I'm just going to do tracking of points. So then I have to define locations or give tags to points on the body of the mice. So let's give it right here. Okay. So this is in the YAML file. So I'm going to left here. It's nose and tail start. Okay, so and then I'm just saving this. So I have defined 4 points. I'm just going to track these 4 points. So here you can construct a skeleton. So in fact, let me just quickly show you. So right here to left here. So let's say left here to nose. So there's a segment going from left ear to nose. So there's a segment going from, let's say, nose to right ear. Okay, so just this is how we define skeletons. So all of this can be done at in the config.yaml file. So that you just edit and you save, and anyway you can just click this and then load this file again. Okay. So you can close the session, open again, and then once you load the config file, you can start from where you left, right? It's very simple. So config file gives you all the information about the state of this DeepLabCut particular project that you're working on. Next is, just like in any deep learning, you have to extract information from, let's say, video. If you want to train on a video, what you usually do in deep learning is you extract a few frames. You want to make sure those frames are disparate enough so that you have a diverse training data set. So this extracted frame will be used to train the network. So there's the training data set, there's the testing data set, and then you can also evaluate that network after this training and testing is done. Correct? That's how you do traditional machine learning. If you're not familiar, please watch the set of videos that I've shared over email. Okay, so this is all very, very vanilla stuff today. So you can do automatic extraction. You can do k-means based extraction, which is to say it will do clustering by just looking at the video, depending on how diverse how similar the image is, and then from that clustering it will pick samples so that you can create a training set which is diverse. So it is done in the background. It's going to take some computation. You can use that, or you can use uniform. Uniform is just going to extract randomly from the video, right? So you can also crop. You can crop with the GUI. I'm not going to do. Once you click clock with GUI, you'll see a small window appears. You just pick the location where you want to crop. So okay. I'm just going to select the same video. It's actually not required. And then you say extract frames. Okay. So frames were successfully extracted for the videos of interest. We'll see the frames that are being extracted. So these are the frames that are extracted from the video we just saw. Okay. So we're going to use these frames to train the network, right? The next step is to label and then train the network. So we go to the next step where you create labels. So I'm going to click label frames, click the folder where the images were selected, and then it's going to pop up this software, which is a separate software called Napari. Napari is just to create a skeleton. In fact, you can, if you know the marker locations, I'll show you. You can do it yourself. But this is just a nice GUI to create labels, right? So this button will help you move the video. This is going to select markers once you place them. This is going to help you add markers. So this point, I can create points, and the point that will be added is shown here. So the first point, as you might remember in the config.yaml file, was the right ear. So you see right ear, left ear, nose, and tail. So those are the 4 points available. So now I'm going to, and there are different ways of labeling. You can do it sequentially, which means it will go from right ear to left ear to nose to tail. Or you can do loop, which will tell you, I'll just mark nose on all the frames. And then I'll do right ear on all the frames, I'll do left ear on all the frames, and then the nose, and then the tail start. Right, so this is just, just an option. You can play with it, and then it's very intuitive. So I have 19 images, so I can actually So this is the tool to move the cursor. So this is the right ear. So let's say I place it here. So next is the left ear and the nose and then the tail. Right, and I can press right key on my keyboard. It will go to the next frame. So this is right, left, nose, tail. Go to the next frame. Right, left, nose, tail. I'm sorry, you have to watch this happen in real time, but but maybe it's worth seeing this, because it's only as much time that's required for you to perform this assignment and for you to do the mini project. Okay, so bear with me. For the people who are going to watch, hopefully we'll edit this out. You can watch the YouTube tutorial that is there. It basically will show you all of what I'm doing today, but without any audio. That's maybe the only USP of the demo part. Okay. Don't worry, I'm not going to make you sit through the training part. I already have a train network, which, like I said, I did this morning. I'll show you how that works. So depending on how big your skeleton is and how bad your data is, the amount of training required might change. Okay, I've reached 19 frames, so maybe it took me a couple of minutes. So it's very important, you have to save it. So just go here, File, Save Layer. Okay, I just clicked it. Then you can close this. Once you do that, you'll see, and this is the folder where the data is. It will create a CSV file and a HY file. Both are just simple, arrays. You can open the CSV file. You'll see the body part, the XY coordinate, image number. That's it. Very, very simple. You don't even need Napari. If you want to do it by yourself, you can do it yourself. It's just a nice GUI to perform this labeling. Okay, so that's the labeling part. The next part is to just create a training data set. It basically tells you which network you want to train on. There are so many options: transfer learning, ImageNet, Super Animal Bird. I don't even know the details of all of that. I just know ImageNet. It's a classical network. I just pick that. Shuffle is just a tag for the particular session that you are working on. You can create multiple projects in the same folder, and you can give different tags. I'm just going to do one, so we'll just stick to this. ResNet 50 is a network architecture. This has to do with the number of layers, how you're doing convolution and whatnot. That's it. So I don't even know what augmentation is. Let's not worry about it. Let's just create getting data set. So it is created. So if you go to this folder where we are playing with, so you'll see that it has developed this, it's created this training data set. Okay. Next step is to train. This is the time-consuming part. The longer you train, the better is your result going to be. So this is a standard number that it recommends. You can train on it and see how well it performs. In the morning, I did 50 epochs, which took me in my laptop 15 minutes, 10 to 15 minutes. Okay, so this is the part I'm going to skip because it's going to take some time. But once you run it, maybe I'll just show just train networks, and so that you can see what the training process is going to look like. Okay, so I'll just click. Let's say 50. So maybe let me just do one. Once I click that, it's going to start training. And you will see once the training is done, it will tell you training is done. By the way, all that we are seeing in GUI can be done with Python commands. I'll just quickly show you how to do that. Yeah, so now the training is done. Next is to evaluate the results. So we'll just do evaluation. See what's happening. Okay. So this evaluation is just comparing the predictions with whatever it was trained on. Okay. The next is to use this trained network on the video that we have added. Yes. So there's filter prediction, which will prevent large jumps in the marker locations or the marker points locations. Just a standard filter, which will just filter such trajectories. You can plot all of them if you want. That's it. Now you click analyze video, which might take a little bit of time. It's just running this network on the full video. Okay. The next step, once it's done, is you can plot the video, the trained data, or the final result. Essentially, you can plot it, and then you can also build a skeleton from the skeleton data and add it to the video. If there are outlier frames, few of them, you can add those frames again, go back to training the network, and then run the new network on your data. Okay, so that's it. It's as simple as that. Once you're done, this is the result you're going to get. I'm really not exaggerating. This is actually what I did this morning on, again, 14 images or something. This is what happened. I just ran it for 50 epochs, 50, right? You can see it's pretty incredible. Especially after filtering, this is this particular video has 3,000 odd images. I'll share this entire folder with you guys. Obviously, 3,000 odd images. It took us less than 10 minutes to do this entire process, and you get successful tracking. And it really speaks to the success of ML in computer vision. Okay. So that's what it is. It will generate the CSV file. Very, very simple. You can see the CSV data of all your body parts. 2,329 images, frames. Okay. Okay, so that's where the demo ends, and that's where we get to the mini project. I think I've gone past my limit. I'm sorry about that. This is the mini project. You're supposed to, you have to shoot a video of an insect or an animal in your neighborhood, in your room, in your corridor, your favorite cat, in your hostel. Use DeepLabCut to track the, let me restate it, not just the insect, the animal. Submit the processed video. Okay. So example videos from the DLC website will not be accepted. Please take the assignment. It's a fun assignment, fun mini project. The incentive is the coolest ones will feature on our website. Maybe the next time we run this workshop, viewers will get featured in the workshop. Okay. And of course, as we have mentioned in the email and also in the poster, certificates will be provided after you complete the mini project. There's one more mini project that Professor Danny will give you. These are not super intense. You don't have to worry. These are not just fun stuff. Okay. The deadline is 17th January. Okay. Please feel free to write to us if you have any questions, guys. We are happy to answer them. I don't want to keep you for any longer. It's already much past what we had aimed for. Thank you for staying back, and see you again tomorrow at 5.